Hi rotators, this is John. I'm a second year in the Gellman group, and welcome to the first part of the virtual lab tour. So in this segment I'm just going to basically walking us through our modules and showing us where all of our instrumentation and some of our storage and just general layout of the labs themselves. So we're going to start off over here. This is Lab A, and coming in through the right side of the door here we've got some of our standard chemical storage, as well as our sonicator over here, which we use to dissolve a lot of our compounds, and our pH probe to measure pH. This is one of our freezers where we typically just store peptides we're working on, or just need storage for peptides that need to be kept at a lower temperature, or any compound we're working on. Over here we got into our refrigerator and our amino acid cabinet. Both of these are just filled with the amino acids that we're typically using in our synthesis to make our peptides, and then weigh station to be working out, to actually weigh them out in. So, and then over here we've got some of the desks that we use. So we've got our bench space, cabinets underneath, storage up top, and then we've got the full hood, usually one per grad student. And pretty much this follows the same general structure for all of our benches and hoods and all of our modules. So coming over here we've got another bench. This one we use for storage or dry cabinet to keep our compounds that we need to keep more away from air and water, as well as our thermocycler here, which we use for PCR amplification or mutagenesis typically. All right. And then coming around this corner, we have one of our HPLCs. We got Big Red and Kate running.
Hi.
And Kate running some samples. Over on the right here, we've got our LCMS. In the back corner over here, we've got our glove box, which we use for compounds that are more air sensitive. And then further...
Actually, I'm wearing a very dorky shirt right now because it's my argon shirt. So you can see there's a map of the synchrotron on the back.
Crystallography team represent.
Yeah.
And then the last thing we've pretty much got in this module is one of our hoods, which we use for storage and waste before we send them out, and also TFA storage, since we use a lot of TFA, especially in our peptide cleavages. And the only other thing I guess I can add is if you're ever looking to find nano-pure water, we keep it in this corner. All right, see you guys in the next segment. Hi everyone, welcome back to the virtual lab tour. And right here, I'm standing into the doorway we walked into Lab A, and across the hall we have Lab B. So I'm going to go through the same sort of thing where I talk about the unique instrumentation of each room, and it's, hey Kate again, and the instrumentation and just general layout of the lab. So as soon as you walk in on the left door of Lab B, you have two machines facing you on the left. So you've got Hobbs, our UPLC, and Calvin, our HPLC. Calvin is more of our workhorse, just like Big Red, and doing the actual menu separations and purifying of the compounds and peptides that we're working with, and collects them neatly in these test tube racks up here. The UPLC does more quantitative breakdown of what you have. So you do all your work on your HPLC, and there may be an artifact or just not as high resolution as the UPLC. So you do a quantitative purity check on Hobbs. So coming to the right of our UPLC and HPLC, we've got our Gyromax here, which we primarily use to just grow up cells and incubate them, that we're going to use for transformation, expressing a protein, or more of our biological purposes in the lab. You walk a little bit further down the alley over here, and now we've got one of our manual peptide synthesis stations. So here's our microwave, which we do our deprotections and our couplings in, and our prep station over here, which is our aspirator, which is used to drain off solvent and reactant material that we have in our syringe that we're using as the reaction vessel. All right, and as we come across into this corner here, we see our safety shower and our eye wash station. So if you ever get exposed to a chemical, get anything in your eyes, it's located right here. And all of our labs have these in a similar location as Lab B, and one of our fire extinguishers located here. Coming down into the middle of the lab, we've got the lane where we have two of our benches and the overall lab stations. So similar to in Lab B, we've got our bench top, cabinet space underneath, shelf space above, and then a whole hood per person working at the station. Now, and then coming over to the last part and the right side of the room, we've got more just lab benches and fume hoods down this alley. A little to the right here, we have our nano drop, which we use to assess assess concentration of peptides and compounds we're working on by absorbance. And then further down here into the last corner, we've got our microscope. And this is primarily used by the crystallography team just to assess and measure and analyze crystal growth as we're growing up our crystals. All right, and I'll see you guys in Lab C in a moment in the next part of the video, for the next part of the tour. Hi everybody, welcome back to the Gellman Virtual Lab Tour, and I'm standing where we walked in on Part B, and now we're going to be walking across to Part Lab C. So, as you walk into Lab C, you'll see that we have chemical storage here with common use things such as PCR tubes and centrifuge tubes that a lot of people use in the lab up here, as well as another chemical storage cabinet here. So, moving forward, we will see that we've got the standard sort of setup here where we've got benchtop, fume hood, and then going to the middle lane, the same sort of setup we've been seeing a lot of, the benchtop, fume hood. But this desk is interesting because no one is currently occupying it, so we've actually set up a apparatus to exclude light for our photosensitive reactions. So coming around the last corner here, we've got Brian weighing out some peptides. Hi, Brian. And the same sort of microwave setup that we have seen in the other lab, Lab B, where microwave as well as chemicals that we use for the actual synthesis and the aspirator to drain off solvent. In the corner over here, we have our Liberty Blue, which is an automated peptide synthesizing machine, which is very handy when you've got a longer sequence to work with. And the last stop we're going to talk about as we go through Lab C is going to be our circular dichroism meter spectrometer over here, Bilbo Baggins, which we use to assess chirality in some of our peptides and helicity in other peptides. So that's about all I've got for Lab C. So I'll see you guys again in our last module, Lab D. Hi everybody, welcome to the final video of the Gellman Virtual Lab Tour. So I'm standing where we walked into Lab C, and I'm going to be going out that door now and heading over to our last lab module to talk about Lab D. So when you walk into Lab D, the first thing you'll see is more chemical storage for our inorganic chemicals. On our right over here, we have our FPLC, which we use to purify out proteins that we've been expressing typically by our cell cultures. At the corner here, we have an empty desk that can be potentially occupied by one of our new rotators that could be joining this year. And another one of our eye wash stations and our fire extinguisher. Coming over to here, we've got the standard setup that I've been showing off in the other labs, which is the fume hood, cabinet space, and desk. And then following by in the next row, the same sort of setup that we see is typical of all of the Shane labs, Shane Tower lab modules. But at the end of here is an instrument I'd like to point out, which is the LIO. The LIO is a device that we use in order to remove as much solvent as possible and much greater amounts than if we were to blow it off with just, say, nitrogen. It works by, inside each one of these jars, there's a sample, and it reduces the pressure, causing a vacuum, and since the vacuum is now in place, the solvent will evaporate easier than if it was at room temperature, for instance, or room pressure. So, the last thing that we have to talk about in Lab D is in the corner over here where we've got one of our rotovaps and a balance, as well as some more chemical storage. All right, and that leaves us finishing up all of the instrumentations in actual lab space. So while I'm here, I'd like to talk about just the general setup that we have for the offices. So all the offices in Shane follow the 3 on one side, 2 on the other. Depending on the office, it may be mirrored, so 3 on this side, 2 on this side. But they become very cozy and will very quickly become your home and very comfortable to sit in since you spend since you're going to be spending a lot of time here anyway. So that brings us to the end of the in-person, virtual walkthrough of all of the instrumentation. We're going to have more videos talking about techniques that we use a lot, as well as instrumentation used in other videos. Well, look forward to seeing all of you as you rotate through, and welcome to grad school.